Jai Hind everyone, I am Pankaj Kumar and today's video is about the polysaccharides. So till now, we studied in our previous videos about the monosaccharide and the oligosaccharides. So if you didn't watch that video, please click the right side corner link which is showing here. And now we'll continue our lecture with the polysaccharide. So it is very important. In this lecture, we are going to read about the heteropolysaccharide, homopolysaccharide, and their uses and their degradations, which can be inhibit the formation of the heteropolysaccharide. So let's begin our class. Average weight of the polysaccharide is around how much? It is around 20 kilo. Okay, 20,000 kilodaltons. So type of the polysaccharide is two type of polysaccharide you can see. Homopolysaccharide. In homopolysaccharide there will be two types, the linear or branch. In homopolysaccharide there will be one single type of unit will be attached either in linear manner or either in a branch manner. So you can see this is a homopolysaccharide. Now other is what? Heteropolysaccharide. In this there will be two or more than two units are attached in a linear way or in a branch way, that is called heteropolysaccharide. As it is shown here, there is a green as well as red balls are available which is attached in a linear manner. So it is called as a linear heteropolysaccharide, and if it is in branch form then it is called branch polysaccharide, heteropolysaccharide. Okay, so let's begin with our first important point, that is called homopolysaccharide. So in homopolysaccharide, we are going to see storage homopolysaccharide and there will be another structural homopolysaccharide. In storage, mostly the storage will be occur in the plants, but is it only occur in the plant only? The storage also required to be occurred in the human beings and the animals. So in case of the plant, the storage will be occur in the form of starch. Okay, it will occur in the form of starch, and in case of animal, the storage will be occur in the form of glycogen. So in case of starch, the unit is what? Glucose. In glycogen also the unit is the glucose only. But in starch, we will find the different two type of structures will be there of the glucose. Like one is the amylose, another is the amylopectin. So in case of the amylose, the glycosidic linkage will be one alpha 1 for glycosidic linkage. There is only one arrow in one direction. It is showing the carbon will be attached in one direction from first carbon to the fourth carbon of the another sugar. In amylopectin, it is showing alpha 1 4. It means it is attaching with the alpha sugar of first carbon of the alpha sugar to the alpha sugar of the fourth carbon of the second sugar. Okay, again I am repeating, alpha means what? It is a sugar, alpha sugar, and first carbon is attached to the alpha sugar of the fourth carbon of the alpha sugar. Okay, so this is a linear form and another is the branching. In amylopectin you will get the branching. Because of this branching, it will getting the alpha 1,6 branching, and at every 24 to 30 residue there will be one branching you will get. Because of this amylopectin branching, amylopectin is not available to bind with the any reducing agent, it is not used to bind with any reducing agent like iodine and all. So it will be not give the taste. Amylopectin is not good for the taste for the starch because it doesn't have a space for binding of the iodine. Instead of that, amylase can use for it. Now glycogen, if you see, glycogen unit is again glucose and it is only linear as well as branched, but it is mostly like to the amylopectin. Okay, so glycogen is mostly like to amylopectin. So there will be branching. There will be branching at every 8 to 12 residues. So each branch of the glycogen end with the one non-reducing sugar. So there will be only one reducing sugar. There will be only one reducing sugar. But how many non-reducing end will be there? So n plus 1 will give you the non-reducing end. Okay. So what is the meaning of n? N is a number of the branches in the sugar. Okay. Now we'll see the structural homopolysaccharide. In case of structural homopolysaccharide, what you'll find, it will be going to be form a structure. Either it will go to form the outer layer like cell wall of the plant, or it can form the exoskeleton of the insects. So in case of the structure cellulose, which having a beta glucose as a unit, and it will be found mostly in the wood stem, or it will find in the fiber wood stems, and it will be found in the every plant cell. We cannot specify only one structure, but it is present in the fiber wood stem. Now it is an insoluble in water, but it having a linear beta 1,4 linkage. It is a linear beta 1,4 linkage, and it is most abundant. This is the important thing. Most abundant biochemical. Okay, most abundant biochemical on earth. Okay, most abundant biochemical on earth. Now we are going to see the another structural organ. Another structural polysaccharide, homopolysaccharide, that is a chitin. If you'll see this chitin, so chitin is the having a one unit which is called the N-acetyl glucosamine. So it is made up of the unit N-acetyl glucosamine, and it will found mostly in the fungi and exoskeleton of the cockroach-like structure. Okay, cockroach-like insects. So beta 1,4 is the linear linkage. It is a beta 1,4 glycosidic linkage, and it is a linear only. So you have to remember it, the chitin and the cellulose. Now stability, if you talk about the stabilities, how they will get the stability, this is the question. Okay, so how the homopolysaccharide will get the stability? What do you think? So mostly the homopolysaccharide will get the stability because of their arrangement of the carbons, carbon. So most stable structure form the helix, mostly helix, like in DNA also, like that the polysaccharide also form a helix for the stability. And the alpha 1,4 linkage chain starts glycogen stabilized by the hydrogen bond. Okay, this is the important lines. Mostly if you will see the amylose, it will have the 60 degree of the angle, turning angles for their stability. There will be from one molecule to the another molecule of the glucose, there will be a turning. Yes or no? There is a turning between a two glucose molecule, and that will be the 60 degree. So iodine test or the starch test, how we can do? By the use of the iodine. As I said, the amylose, the core of the helix is of a precisely right dimension to accommodate the iodine. So it is having a precisely right dimension to accommodate what? To accommodate iodine. Okay. So iodine like I3, I5 is the goes and to accommodate, and it will give the most important color, that is the blue color. Okay, it will give you the blue color if the starch or the amylose will be there in the sugar. Then now we are going to read about the heteropolysaccharide. So heteropolysaccharide, polysaccharide. So what we are going to read about the heteropolysaccharide, the most important thing about the heteropolysaccharide is that it having a different unit. Now the heteropolysaccharide also form a structural unit, or it will form a like a complete matrix, or the structure, as well as not exactly storage, but as a communicating signaling molecules and all these things. So this is the very important things. So we are going to start with the peptidoglycan. First is what we are going to start with the peptidoglycan. Peptidoglycan. So see, if peptidoglycan, if you'll see, there will be two unit in the. There should be two unit in this. So what are the two units? So the two unit is what? First and two units. First and the second unit. The first unit is the N-acetyl glucosamine. What it is? N-acetyl glucosamine. And another unit is what? N-acetyl muramic acid. But the question is that are they form the branches or it will be in the chain? What do you think? You can comment it here. Acetyl. Here it is one L will be there. Acetyl muramic acid. So N-acetyl muramic acid and N-acetyl glucosamine are the linear in form. Which form? They are in linear form, and if they are in linear form, there will be one bond. Which bond? There will be glycosidic bond between them, and that is a beta 1 for glycosidic bond. All are the glycosidic bond in the carbohydrate. So I am writing here glycosidic bond. Important to remember here, the penicillin, which is extracted from the penicillium notatum. Okay, so the penicillin, which is a antibiotic, and it will prevent the cross linking between the prevent peptidoglycan cross linking. Okay, peptidoglycan cross link. Prevent peptidoglycan cross link. Hence bacteria die. So bacteria will die because of osmosis, osmotic lysis. So we were seeing the heteropolysaccharide, like a peptidoglycan, is the cell wall of the prokaryotes. So if it is cell wall of the prokaryote and it have a unit of N-acetyl glucosamine and N-acetyl muramic acid, so that linkage beta 1 for glycosidic linkage can be blocked. Okay, this linkage can be blocked by the penicillin. So the cell will die with the osmotic lysis. So another is the GAG, glycosaminoglycan, which is very important, and it is present mostly in the matrix. It is present mostly in the extracellular matrix of the body. So what is the purpose of it? It will help in the diffusion of the nutrient and the gases between the tissues, between the blood capillaries, between the lymphatic tissues and all. Now the question is that, is there only sugar or GAG in the extracellular matrix, or is this sufficient about the sugar and the sugar to help in the diffusion in all these materials? So the answer is no. No, it is not. So there will be some another molecules as well. So what are these another molecules? So another molecules will be what? The fibronectin and the collagen and the elastin fibrillars, all are this extracellular matrix component which are protein in nature. Which are protein in nature. Got it? So these are all protein in nature, and it will help in the giving the strength and the shape of the tissue and helping in the communication between the two tissues as well, and give the elasticity and all these things. Now we are going to see the units of the gag. What are the unit of the gag? As you know, the gag is a heteropolysaccharide, so there should be two different unit. At least there should be two different unit. So the first unit is what? The N-acetyl glucosamine. What it is? Either it will have the acetyl glucosamine, or it will have N-acetyl galactosamine, or you can write here, or N-acetyl galactosamine. For better experience of this video, you can fast forward the classes whenever it is little going slow. Okay, so whenever I'm writing, it will take times. So acetyl glac uh, galactosamine. So this is the what? Gag unit. This is the first unit, but where is the second unit and what it is? So the second unit is what? The second unit is the either it is a not either it is a uronic acid only. Okay, it is a uronic acid, and there will be two type of uronic acid. It will consist either it will have the D glucuronic acid, uronic acid, or it will have L hydronic acid. L hydronic acid. Okay. Now the question is that, sir, which type, Pankaj, which type of the bond it will have? So the bond it will have different type. Either it will have the alpha 1,4 glycosidic bond, or it will have the beta 1,3 glycosidic bond, or it will have the beta 1,4 glycosidic bond. Why I'm saying this many type of bonds can be possible, because sometimes it will have the N-acetyl glucosamine with the D-glucuronic acid, or sometimes it will have the L-hydronic acid with the N-acetyl glucosamine. So it can be vary, but these three type of the bonds can be seen in the gag. Now, if you see the gag have the more advanced structure. If the uronic acid will bind with the if uronic acid, sometime it will bind with the sulfate. Okay, so it is binding with the sulfate, and it will giving the extracellular matrix a big potential. Okay, so sulfate, actually the sulfate will bind with the COH group of the gag. Okay, of gag, it will bind usually with the carboxylic acid group of the gag. What it is giving this? It is giving the sulfate is there now. So whenever the sulfate will come, the sulfate will give the negative charge. Okay, give the negative charge density. So if it is giving negative charge density, then what will be happen? If it is giving a negative charge density, to minimize this negative charge density, it extend to form a rod shaped helical structure, as like in the heparin. Okay, so this is a negative charge now. It is, suppose this is a small negative charge, there will be so many negative charge nearby this. So what it is happening, it is increasing and making a rod like structure, complete rod like structure. So the negative charge will be far apart, so there will be less repulsion in this. So it is forming what? The rod shape structure. So what you can write, to minimize negative charge, it forms rod like structure. Got it? So as you can see in this diagram as also, rod like wait, rod like helical structure you can write. Okay, helical structure. Now I am going to show you the picture here. So it forms this type of structure which is represented here. That is a heparin segment. So this type of structure you will get here in the if the sulfate is binding. So in if the sulfate will bind with the GAG, it will form a different type of structure. Either it will form the hyaluron, either it will form the chondroitin, either it will form the keratin, or either it will form the heparin. So we are going to read all these four. So uh, hyl- hyaluronan is a simple sugar which is glucosaminoglycan and N-acetyl glucosamine. Okay, so it having a beta 1,4 glycosidic bond, and this picture was taken from the beta 1,4. Let me read. So this picture was taken from the Leninger book. Okay, beta 1,4 glycosidic bond. So now if you add the sulfate, okay, if you add the sulfate in this, then it will become what? Chondroitin. If you will add the sulfate on the another carbon of this N-acetyl glucosamine, then it will become the keratin. You can see this sulfate, the changes in the sulfate, position of the sulfate. You can see here. Other first time it is bind here, second it is bind with the another carbon, third it is forming here three sides. Okay, so this is heparin. It is making a complete new differences in the sulfate. But before going to this, all the structure, we just want to write the simple proteoglycan. So whenever the sulfated glucose is attached with the ECM extracellular matrix protein, then it will form a proteoglycan. Let's begin with the first point, that is a hyaluron. Hyaluron. So what are the point you have to mention? Remember in this class. So hyaluron unit is what? D-glucuronic acid and N-acetyl glucosamine. So its unit is what? D-glucuronic acid. D-glucuronic acid and N-acetyl glucosamine. N-acetyl glucosamine. So here you can see in this the bond you will get beta 1,4 glycosidic bond you will get in this. Okay, it will serve as a highly viscous non-compressible solution lubricant in the synovial joint. So it will present in the synovial joint, as well as it will present in the vitreous humor. Where is the vitreous humor will present? So vitreous humor in our eyes. So it is a lubricant and it is non-compressible solution. Important thing about it, its size is around 50,000, of Dalton. Okay, so this is about hyaluron. Now we are going to see hyaluron is, now we will see the chondroitin. Before going to the chondroitin, I am going to mention some more point. Hyaluronan is a component of the cartilage, and the result of the non-covalent bond provide the strength to the cartilage. So it will present in the cartilage also and provide the strength, provide strength and elasticity. One more places you already heard this enzyme. It form an enzyme that is a hyaluronidase. You remembered it? Where it will be present? Hyaluronidase. So usually it will present in the sperm head. Sperm head, specifically in the cap. So this is sperm head, which is present in the cap of the sperm. it will help in the penetrating the ovum. So the gag which is surrounding the ovum, that will be penetrated by this hyaluronidase. Okay. Now we are going to see the next, which is called chondroitin. Okay. So if we'll see the chondroitin, so chondroitin is here it is. Okay. So chondroitin having beta 13 and beta 14 glycosidic bond. Same like this. Beta 13 and beta 14 glycosidic bond. But here you will get the 1 sulfate group. So what is the size? It is around 20 to 60 unit. So chondroitin will help present. It will found in the cartilage, tendon, ligament and all. Okay. Before that, you just write it is shorter, 20 to 60 unit, and it will present in cartilage, ligament and tendon. So whatever the function you are seeing in the tendon and ligament, that will be provided by the chondroitin, and the same function will be performed by the heart valves. Another is what? Dermatin sulfate. Okay. Now we are going to see dermatin sulfate. Dermatin sulfate. Both spellings are correct. PHAT and SFAT. Both are correct. Okay. So it is shorter. You can see this dermatin sulfate is not mentioned here, but okay, fine. So it is shorter than the chondroitin. It is shortest than the chondroitin, and it will provide present in the skin, and it will give the pliability. Okay, it will give pliability, and also present in the heart vessels. Also present in the blood vessels. Okay, blood vessels. Now the last, the keratin and then heparin. So the keratin, you all knows from the childhood, keratin sulfate. So this is a modification just because of the adding of the sulfate group in the gag. If you add the sulfate group in the gag, these many type of the things will be formed. Just keratin if will form, remember it, if the keratin will be formed, there will be no uronic acid. No uronic acid. Only N-acetyl glucosamine will be there, and there will be no uronic acid. Now, come to the point. So where it will present? They present in the cornea. They present in the cornea, bones, like horny structure. What is the horny structure? Wherever the structure, like a horn-like structure. Now you can write nails, hair, hoof, and all these things. Okay, and claws. So this type of structure it will be there. Now the heparin sulfate. Okay, already you heard this heparin. Can you guess where it is present in our body? Heparin sulfate. So the heparin sulfate will be present in your heparin. In the basophil cell, the histamine will release. and the not in basophil, actually in the blood the heparin will be there, which will be anticoagulating. Okay, good. So first it is isolated from the dog liver, and it contain variable random arrangement of the sulfate and non-sulfate. So contain variable non-random random arrangement of sulfate and non-sulfate sugar. Sulfate sugar. Got it. Very nice. Now the function will be what? You know, it will be present in the heparin released from the mast cell again. So mast cell release what? Heparin, and it will present in our blood, which will help in the anticoagulation. Okay, anticoagulation. It will also participate in the growth factors. Okay, interact with the large, interact with growth factor. So thanks for watching this video. If you want to solve some questions, please go and solve the questions which is given in the description. I am giving doing hard work for you as well as for myself also. So I'm giving you this question. Like this question, you will get more in the description box in the U Biology app. You go and download that, and now you solve this question. So now you have to find the incorrect answer, incorrect statement among this. Okay, so choose the incorrect statement. You just pause your video and find the answer. So if you seen properly, there is a one mistake in this. This is a alpha 1,6 linear. It is written. Okay, or it is written linear, but alpha 1,6 is a branch, so it should be branched here, and this is a wrong statement. Remaining are the correct only. Like that, there will be some practice questions, and there will be some analytical questions which is also asked in the CSIR June recent exams. So we are giving the analytical questions as well as. So before leaving this video, this is a very important chart. You can copy and you remembered it. Like whatever we studied till now, this is concluded in this table. So starch will have the amylose and amylopectin, and both are homo. This type of bonds will be there. These are the units. And glycogen, homopolysaccharide, alpha 1 for glycosidic bond, alpha 1 6 branch, at 8 to 12 residues. The branches will be available. Like cellulose keratin, these are the structural units, and it have a beta 1 for glycosidic bond, and acetyl glucosamine will be there. Then dextran, homopolysaccharide, alpha 1 6 glycosidic bond with alpha 1 3 branches will be there. Wide range structural in bacteria and extracellular adhesive. Peptidoglycan, as you seen, hyaluronan, as you seen, GAG, beta 1 3 and beta 1 4. Okay, 8. So these are all about the bonds and all. I hope you enjoyed this video. Please do solve these questions, and we'll meet in the next question, next meet class. Thanks for watching. Jai Hind.